Good afternoon and welcome to this Orkney International Science Festival event online. My name is Heather Woodbridge and it is my pleasure to be hosting this session from Northern Isles in Orkney this afternoon. The Orkney International Science Festival is entering its 30th anniversary this year, and we are marking the occasion with our very first online festival. This year, we are delighted to bring a full programme of events directly to you wherever you are in this moment. Well, today I'm joined here by Dr. Shona Kerr. Hello, Shona.
Hi.
Dr. Shona Kerr is from the Medical Research Council of Human Genetics at the University of Edinburgh. Shona's degrees are from the University of Glasgow and King's College London, and she has had a long research career in Cambridge, Oxford and Edinburgh. For the past 4 years, she's been working with Professor Jim Wilson on the Viking Genes project, the subject of today's event. Well, Shona, please tell us more.
Delighted to do so. Okay, great. First of all, thank you so much to the organisers of the Orkney International Science Festival for inviting me to give this talk. You'll see there the title, Viking Genes. And if I could have the next slide. This is the synopsis of the talk that I agreed with Howie Firth back in February. And we thought it would be of some interest, to tell people about our new project, our new recruitment project, which has a target of finding 4,000 people worldwide who have at least 2 immediate grandparents from Orkney or Shetland. And the reason we want to do this is to look at the genetics and the health of these volunteers. We want to use this to better understand what might cause diseases such as heart and eye problems, diabetes, cancer, and really any other common complex condition that has a genetic component. And so we agreed I would provide some of the methods we're using to achieve this purpose, and give some updates on the project. And other than the fact that it's not possible to come along and hear from me, we're making use of the marvels of digital media to speak to you today. Next slide, please. So, first of all, why did we pick the Northern Isles for this genetic study? This work has been going on for a long time, and it's clear that genetic analysis of a sampling of populations across the whole of the British Isles and Ireland shows that genetically, Orkney and Shetland are the most divergent and isolated populations of all. The figure on the right here is a sort of tree of relatedness of populations. Down at the bottom, you've got England, which can only be resolved into really a very small number of separate groupings. Similarly, Wales. There's a little bit more differentiation within populations in Scotland and Ireland, but you can see that in both Orkney and Shetland at the top, it's actually possible genetically to distinguish people with ancestry even from individual islands within Orkney or Shetland. And that's really of some interest for understanding the genetic landscape of the British Isles and Ireland, and also genetic ancestry within the various regions. But it also means that there's potential for there to be a different pool of medically relevant gene variants, which would be in Shetland and Orkney at different frequencies compared to mainland Scotland. And the work in the tree that I've just shown you is published in a paper that came out last year, which is really the culmination of 10 or 15 years of work led by Jim Wilson. And this paper that we published got really quite a lot of publicity at the time. And you can still read about it on the BBC News website. One of the other findings from this comparative work is that the Shetland and Orkney have the highest proportion of Norse ancestry in the whole of the British Isles. And that's why we gave our project the overall title of Viking Genes. So as I've said, Orkney and Shetland have special populations. There's strong and deep family relationships and unique shared ancestry. And this has been known really for some considerable time. This slide shows the very first research paper identifying a genetic variant, in this case, in a population from the island of Westray. And it's a variant in an enzyme found in the red cells in the blood. So that paper was published all the way back in 1972, which is quite a long time ago. But as the next slide shows, maybe not so far back for some of us than others, that's actually me there on my only so far visit to the Orkney Islands in the 1970s. That's me rocking the tank top and polo neck on the beach look. And this slide, which my dad took, and several others, such as in the next slide, that's my dad's car being loaded onto the Orkney ferry at Scrabster. And we've made good use of these antique slides on our Facebook page. And I'm going to come back and tell you a little bit more about our use of social media in the project towards the end. So that's the 1970s. What about the intervening period? Well, this research has been led by Professor Jim Flett-Wilson of the University of Edinburgh. Jim's an Orcadian. Many of you will probably know him or know of him. And he and the team recruited more than 4,000 volunteers to two different research studies, Orcades in Orkney and the Viking Health Study in Shetland. And collectively, we were calling those Viking. These studies required volunteers to attend clinic visits in either Lerwick in Shetland or Kirkwall or the Northern Islands of Orkney. And these recruitment involving clinic visits ended back in 2015. And the eligibility for both of these studies was to have 3 or 4 grandparents from either Orkney or Shetland. In the next slide, I've just pasted in a screenshot from our Viking Genes website. The cohorts have supported really hundreds of research findings and published papers, many of these as part of worldwide partnerships. And we've put some summaries of these pieces of work on our website, which is given there. And as I mentioned, there are these special features of the Viking cohorts. They're family-based, geographically isolated populations. Because of the clinic visits, it was possible to take a number of samples from the volunteers, and these are available for further research and analysis. It's also possible to make use of electronic health records of NHS data, such as hospitalisations, to follow the volunteers as they go on through life. And another useful special feature is that the volunteers can be recontacted and asked if they're interested in taking part in extra add-on studies. And finally, a theme I'm going to develop in this talk is that the Viking cohorts provide increased power to investigate gene changes that are not common in other populations, so so-called genetic rare variants. And before I go too much further, in the next slide, I just want to briefly outline some of the features of genetic analysis. There's lots of different ways of analysing, of doing genetics research, but increasingly it's a high-tech world, and it's possible to sequence the entire genetic material of an individual. So this DNA sequencing, I say it's high-tech, that's one of the facilities we have here in Edinburgh, shown in the photograph. So DNA, the genetic material, is built up of long strings of letters or nucleotides, abbreviated A, T, G, or C. And in DNA sequencing, we effectively read those long strings. And it's worth doing this because the DNA contains the genes, the units of inheritance, which in turn encode for proteins. I think we've got a couple of bullets to come. Yeah, here we go. Sequencing is reading the letters on the strand, and then genes are in the DNA and are a recipe for proteins. And an important point is that it's the variations in DNA sequence that we all have that make us unique and really contribute to all our measurable biological traits, whether height or BMI or all sorts of other measurable traits. And these changes that arise in the DNA sequence over the generations usually have no effect at all, but sometimes if they land in an important part of a gene, they can either increase or decrease the risk of specific diseases. So the next slide gives one example of this. One of the measures that was taken in the clinics was an electrocardiograph, an ECG. And the photos there show the team of clinic staff at the Shetland clinic. And I think the volunteer below is just reading up about what she has to do before getting her ECG taken. And the diagram on the right there shows a trace from the ECG, which is a record of the rhythm and electrical activity of the heart. And in fact, we use that slightly iconic image in all our logos for the Viking studies. And what it's possible to do is to pinpoint each part of each heartbeat. And as illustrated here, these can be marked as P, Q, R, S, and T. And that then allows a number of intervals to be defined. And I'm going to tell you a bit more about the QTC interval. And there's software which allows these intervals to be extracted from the ECG, and then the data can be analysed. And what we've done here is to plot the distribution of that measure, the QTC interval, in the population of our volunteers in the Shetland study. And you can see it's this approximately bell-shaped normal population distribution curve. The x-axis is the QTC measured in milliseconds. The lowest is somewhere below 350, and then there's a tail going up above 500 milliseconds. And it's been known for a long time that these normal population distribution curves apply to very many other measured traits, including other intervals on the ECG, and also things like height, BMI, and even some measures that can be analysed by questionnaire, such as food preference. And this quantitative distribution allows a whole range of complex statistical genetics analyses, one of the most popular of which is GWAS, or genome-wide association studies. And they make use of common variants in the populations contributing their data. And in this sort of GWAS study, the analysis is usually done in combination with data from other populations, and data from essentially every volunteer in the study can be used. So that's the distribution. And then once the genetic analysis has been done, all sorts of findings can be made. This is one paper that was published just a couple of months ago. Jim is a co-author. You'll see there's a very large number of co-authors, and that reflects the large number of cohorts that took part in the study, this multi-ancestry GWAS of an ECG trait. And that was able to identify more than 200 loci, which approximates to genes, which in some way underlie the cardiac conduction. So there's been lots of studies of this type, making use of data from the Viking cohorts. However, I wanted to also give you an example that's specifically from the Northern Isles, again looking at ECG measurement. And we say a bit more about this project on the Viking Genes website if you're interested to learn more. So there's been an absolute explosion of genetics and DNA sequencing worldwide, and there's in fact a database of DNA sequences from 180,000 anonymized, de-identified people, which you can look up for any variant of interest. And this worldwide database of 180,000 people just had 2 instances of a rare variant, which was known for some time to NHS clinicians. And we also found it in the Viking Health study with 5 people with ancestry from Shetland. 3 of those people are in the family A, which I've illustrated above. So this is a fairly simple pedigree. Males are in squares, females are in circles. And the people who are volunteers in our Viking study are coloured in green. And the 3 people who carry this particular rare variant, which affects heart rhythm, are coloured in black, and their QTC value is shown below the symbols. So this pedigree was known to NHS clinicians, and we were able to find it in our research volunteers. But we had more genetic data than the clinicians have available to them. So we were able to do some additional analysis as indicated on the next slide. So here is a visualization, not just of the rare variant, which is indicated by the arrow, but also the patterns of variation along the DNA, along the chromosomes around that rare variant. And it's maybe a bit hard to see, but if you look more closely, in the next part, I've outlined in red the 3 patterns that are common to the 3 people that have that rare variant. And so we were able to look for that pattern across the rest of the 2,000 people in our Shetland cohort. And doing that, we found 2 more people who had the same pattern, the same haplotype as illustrated here. We called this family B. And as I say, 2 more instances of the rare variant, which we confirmed by DNA sequencing. And what was particularly interesting about this was that it wasn't known to the NHS geneticists. On the next slide, we found that this family B was not connected to family A in the recorded pedigree, or even in the excellent genealogy that's available on people from Shetland. And a bit of further digging showed that there was indeed a distant relationship from a common ancestor. But this was sufficiently distant that it wouldn't have been identifiable by the normal method of cascade testing out from the initial family in an NHS clinical setting. So we had this information that we thought would be valuable, and so we were able to return it via the GPs of the participants based on the ECG data. And we wrote this work up in a paper that was published last year, an actionable KCNH2, that's the gene variant detected by sequence and haplotype analysis in a population research cohort. And before I go any further, I just want to define what I mean here by actionable. I think that should come up. Just next. Yes, here we go. So an actionable genetic finding is simply a gene change that is linked to a condition or disease that can be prevented or improved by treatment. And I'll go into this a bit more in the next slide. Yeah, so actionable genetic findings in research volunteers. We would only be interested in acting on these findings if there was a potential benefit to the participant. That's to say that a valid approach must exist to prevent the condition or disease of concern. And also that early knowledge of the genetic risk could enhance the efficacy of that prevention or cure. And we're only really interested in significant diseases or conditions. And the validity, the scientific validity and the verification of the result is very important. And that's why it's really important that we're working hand in hand together with our clinical colleagues. And another key point is that before any results are returned to any research participant, what this means has to be explained and their consent has to be sought. And actionable findings in research aren't just genetics. There's a whole other range of findings can be made which might be useful to the research volunteer. I think the next illustration gives an example from an imaging study where Kermit the Frog doesn't know whether or not he wants to get a potentially important result from an X-ray. So in the next slide, we're not alone in having these interests. Iceland is another very interesting population genetically, and in effect, the whole nation is a population isolate, and they've also got some genes that have risen in frequency in the population. And it's very clear that without explicit consent, neither the government nor any industry can notify people that they might have a higher risk of a particular disease. And so that's the legal position. Not everyone actually agrees with that. Next, please. And we have some similarities to this situation in Iceland, also in the Northern Isles. Next, please. So this is broadly similar potential for a unique pool of clinically relevant variants. But I wouldn't want to give the impression that it's all doom and gloom because of these rare genetic variants. In actual fact, the government statistics usually put people in the Orkney Islands very close or at the top, as in this table, of the life expectancy data within Scotland. And you can see Shetland is also above the average for the whole of Scotland, for both males and females. The error bars, the confidence intervals in both Shetland and Orkney are higher simply because of the smaller number of people in those council areas. And I think you can also see right at the bottom, dear old Glasgow City propping up the rest. And another way of interpreting the data, which I think is quite illuminating, next please, is that it basically translates such that in the Orkney Islands, 1 in 4 boys born 2014 to 2016 could expect to live to be 90 or more, whereas only 1 in 10 boys born in Glasgow would expect to reach that age. So it's by no means just genes that matter. Obviously, environment and all sorts of other factors affect health and longevity. So inspired by all this work, we've recently been able to get more DNA sequence data. We now have DNA sequence data from all of the participants in the Viking study. And we're really just starting to delve deeply down into that. And one of our new areas of focus is in a rare variant found in some people of Westray descent. And Westray is, I think it's fair to say, famous because of the finding of a 5,000-year-old figurine on the island. And this representation of the human form has been dubbed the Westray wife. And she's on show at the Westray Heritage Centre. So our early results show the presence of a variant in a gene called BRCA1 that can increase the risk of getting breast cancer. So we're currently investigating with clinical genetics colleagues whether in the future it might be appropriate to offer population genetic screening through the NHS. And if this did happen, the genetic screen would be supplementary to the existing screens for breast cancer that are available now, such as mammography. And I think quite an important point to make is that the research we're doing does have findings that are relevant beyond Orkney. And that's partly due to the emigration to the New World and antipodes that have taken place in the last few hundred years. So building on all these findings, we were able to launch a new study, a new recruitment to a study called VIKING II, which is funded by the good people at the Medical Research Council. And again, a screenshot from the Viking Genes website. The VIKING II study on the next slide is an online study. So unlike the previous cohorts, there's no clinics. And we launched full recruitment back in January 2020. I must say it feels a lot more than 8 months ago. Such a strange year this has been. So it's an online study with an online questionnaire. And but because we're interested in the genetics, somehow or another, we have to get hold of DNA. It's possible to collect good quality DNA from saliva samples. So we're using a spit and post method to collect DNA samples from the research volunteers. And for the first time, we're taking direct consent for return of selected actionable genetic results with the NHS. We had to pause sampling due to the pandemic, but we're back in business now as of a couple of weeks ago. Next slide. And another key point about VIKING II, next please, is that the eligibility is 2 or more immediate grandparents from Orkney or Shetland. And because it's online, this means that participants can be anywhere worldwide so long as they fit the eligibility criteria. Next, please. And next again. So here's some evidence that it's a worldwide study. This talk is in the Orkney International Science Festival. So of course I've started with Orkney. Each of these pins represents the approximate location of a volunteer in VIKING II. And you can see the great numbers we're getting in Orkney. Next, we zoom out a little to also include Shetland. And in fact, 44% of the participants at the moment are from the Northern Isles. Zooming out further, we can see that lots of other people in Scotland have participated, 37%, and 11% in the rest of the UK, England, Wales, and Northern Ireland. And then the rest of the world, we're up at 8% now. Here's our world chart. And you can see there's individuals pretty much everywhere. But it's really interesting, I think, to see that we still have really good numbers in Canada, mainly people of Orkney descent, and also good numbers in New Zealand and the east coast of Australia, and they're primarily of Shetland descent, reflecting the old migration routes from the past hundreds of years. So that's where we're at. The next slide just tells you a little bit more about the spit and post sampling. That's gone really well. That's our kit there with its return stamp. Normally works smoothly. The dog in that instance didn't quite eat the homework, but ate the packet. But normally, next, it all goes quite smoothly. People have followed the instructions beautifully and posted the kits back to the lab in Edinburgh. Next, please. And people in the UK can just use the stamp that's on the packet, but volunteers living outside the UK have to pay for return postage to Edinburgh. So we're particularly grateful that people haven't just given up their time, but they've also paid to return their sample, which is terrific. And the receiving lab is gradually building up a nice collection of stamps. So the samples go back to Edinburgh. Next, please. And on the left here, we have Kirsty Morris with a big stack of empty packets. The numbers really have been flowing in, which is great. And the lab, the Clinical Research Facility lab in Edinburgh, has been able to work on COVID research throughout the lockdown period. So our project has been a bit delayed, but Kirsty has still been able to book in the samples and keep them safe. and the DNA extractions are now just beginning. Once the process of extracting DNA from the saliva samples is complete and the quality has been checked, the next step is for the samples for the DNA to be sequenced. And in doing this, we're doing this in collaboration with Regeneron, who have a really state-of-the-art DNA sequencing pipeline that ourselves and a number of other cohorts are making use of. And this collaboration model leads to discoveries, and we hope that these will in the future lead to improved patient care. So that's happening in the relatively near future. The next slide, I mentioned that we're planning to return results in Viking II. We've tried to explain what that means. This is a screenshot from our participant information sheet, which gives some information on what we're planning to do and why. And in the next slide, a key point I've already mentioned is that this is in partnership with clinical genetics colleagues. This picture shows Professor Zosia Mead-Brodska in our office in Edinburgh before the days of social distancing. And Zosia is also our independent genetic advisor. And the next slide shows Jim Wilson together with Dr. John Dean having a meeting up in Aberdeen. The next slide is really pretty much wrapping it up now. I've given you a bit of a whistle-stop tour of the Viking Genes project, describing the population cohorts for genetic studies of health-related measures. I've told you a little bit about the types of genetic analysis that can be done, including DNA sequencing. I've outlined our plans for returning limited actionable results to participants with their consent and with the NHS. And finally, I've shown some percentages and some maps, but here are the actual numbers so far in Viking II. We've had a fantastic response with more than 4,000 people registered. But actually, we'd like more than 4,000 people to get right through the study and return samples. So we've still got a bit to go, and we're actively seeking additional eligible volunteers for the Viking II study. So the next slide, very much thanks to the Viking Genes team. Rachel Edwards deals with all the social media and publicity and PR for the project and comms in general. David Buchanan is the data manager and has designed and implemented the questionnaire and deals with all the data management, keeping it safe and secure. And Professor Jim Wilson leads the study. I'd also like to thank NHS Grampian Clinical Genetics and the lab, the Clinical Research Facility in Edinburgh. And of course, very grateful thanks to the volunteers, without which none of this research would be possible. And I promised I'd say a little bit more about our social media. These are the addresses of our Facebook, Twitter, and Instagram accounts. You can also find those on our website and emails that we might send you. And finally, in the last slide, the Facebook page has been particularly popular and a really great way of keeping in touch with our volunteers who like that sort of thing. We've got a fun competition for Friday fun. We've got a couple of ancient bird reports. If anyone's interested, they can get in touch and make the case for getting them. And so we've put all sorts of things out on the Facebook page. Next slide is the ever popular League of Surnames, with indicating some of the Orkney prevalent surnames in the ORCADE study. And finally, what are we going to do next? Well, as I've just mentioned, we're sort of in the process of relaunching. Now that we've come through at least the first lockdown, and anyone who's interested can register interest on the Viking Genes website and get more information. Another piece of work we're planning to do is to go back to the original Viking volunteers and request new consent from them. And once we have the DNA sequence information, we plan to start returning the first results, but that's still some way down the line. And of course, the data that's being collected allows lots and lots more research to be done. So that's all I want to say for today. Thank you so much for listening.
Thank you so much, Shona. That was fascinating. And I think, I have to say a lot of interest, if you're, someone living in Orkney and Shetland. Excellent. And that spit and post, that must have been a huge boost to you at the current situation. That was obviously planned before. I mean, what a stroke of luck.
Yes, it's true. It's true. Yes.
Gosh. Well, we've had a flood of questions pouring in, and we've got a little bit of time to have a look through them. And I'll just ask you our first question, Shona, if that's okay.
Of course.
We've got a question here from Richard Oakley, and he is asking, was a sample size of 4,000 decided upon to give statistical significance?
Oh, that's a good question. Yes, but it was also based on what could realistically be collected given the size of the population. It's actually very hard to estimate how many people there are in the world with 2 or more grandparents from Orkney or Shetland. It's not a statistic that's really known. And so it was a mixture of statistical significance, but also what was feasible and affordable. And as I mentioned, a lot of the research we do isn't just using these cohorts alone, it's in collaboration with other studies to increase the power.
Apologies there, technical hiccup, couldn't unmute myself. Okay, thank you very much, Shona. We've got a question now from Bill Graham, and he's asking, Shona, what does the QTC value mean in terms of heart performance and health? It's a good question, Bill.
That is a good question. So I'm not a cardiologist, but I did show the distribution, and as with many of these things, it's not particularly good to be at the extremes. So a really short QTC isn't particularly good news, and nor is a really long QTC. But there are certain things that that increase the QTC. There's a few drugs that have an effect of increasing it. So if you have a long QTC interval, there's certain drugs it's not wise to take, for example.
Gosh, fascinating. Thank you. Excellent. Okay, we've got another question from Richard here, and he's asking, has this analysis revealed when the Orkney and Shetland Isles were first settled? What can you tell us about the movements of people with this data?
I think that's one to lob to Jim Wilson. And so I think I really shouldn't even try to answer that. I do remember asking him if the sort of Neolithic people had left traces in the modern bloodline, and I believe there's a small amount of evidence for that, but a lot of the data on this is very, very new. It's only recently been technically possible to look at ancient genomes to be able to recover enough DNA from skeletons and preserved remains from the ancient peoples. So I actually think the next maybe 5 years is going to be really fascinating for that kind of genetic archaeology, if you can call it that.
Oh, excellent. Looking forward to hearing more about that. Excellent. Okay, we've got a few more questions, and we still have a little bit of time. We've got a question here now from Paul Cooper, and he is asking, is there a firewall between the information you collect and potentially going to insurance companies? For example, making people uninsurable?
Yeah, so that is another good question, and it's something we have to think about. And in the United Kingdom, it's pretty good. The insurance companies have agreed a moratorium not to make use of genetic information in life insurance or other forms of insurance. And I think that's in everyone's interest. It's actually quite complicated. Insurance companies like to know the average. They base all their statistics on averages. And if people who have, for example, a really great set of genes were to start not buying insurance, that wouldn't be good for the insurance industry. So it's not just about people who've maybe got risk factors buying too much insurance. It's also healthy people not buying enough insurance. So it's actually quite agreed by all that the way to go is not to know about the genetic information. Research data is vital to make sure that it's secure, and we've really gone to great lengths to separate the personal information from the rest of the data. And the only way we would have to divulge to an outside party would be if there was a court order. Mm. And it's very, very rare indeed for that to happen. So, yeah.
So everything you've collected is really very secure?
I'd like to say so, yes. I'm confident I can say that, yes.
Oh, great. Thank you for that. That was fascinating. Okay, we've got a couple more here. We've got another question from Richard, and he's asking, he's saying, you've talked a little bit about the occurrence of disease and its genetic links, but have you found any benefits of having Viking genes? There must be many.
Yes, yeah, I deliberately put in the life expectancy chart, and I believe even back in the day when the Hudson Bay Company was recruiting men from Orkney, it was because they were considered to be so fit and healthy and hardworking. So I think that's quite a hard thing to get to the bottom of genetically. But I do have colleagues at Edinburgh University who are making a big study of longevity. I think it's a really interesting topic. Why is it that some people, it does tend to run in families. Often if your parents have lived to a good old age, then that carries on. But equally, if our modern lifestyle of obesity and lack of healthy living, well, none of us will get to the 90 plus that we would all aim for. So it's not just the genes.
Balance of lifestyle as well is really important.
Yes.
Excellent. We've got a quite similar question here from Jane Harris, and she's saying, does being born in Orkney have more or less influence on longevity? For example, having 3 or 4 Orkney-born grandparents. So I think she's, is living in Orkney as important as having that genetic heritage?
That's a really good question, and actually for the first time we might be able to address that in our Viking II study, because we could look at different parameters in the participants living overseas versus those still in the Northern Isles. I think it's almost certainly fair to say that in Orkney there's a healthier environment than, for example, Glasgow with its air pollution and various toxins still kicking around from back in the day. Absolutely.
Thank you very much. Great. Well, we've just got time for a couple last questions. We've got a comment here from Richard asking, do you need a dog to be in the survey, or is it only if you want it to eat your test kits?
We like dogs in the Viking team. We're always happy to see pictures of pets, but no, the dog DNA would just be too complicated.
Right, absolutely. And just on rounding off on a final question, it's a really nice one to end on, I think. How long will the survey last, and when do you anticipate having your results for Viking II?
Two good questions there. So we're funded by the good people at the MRC until the end of March 2023. But the sooner we can hit our target of 4,000 people with questionnaire data and samples, the sooner we can crack on and get it analysed. So difficult one to answer, but we're anxious to progress as much as the volunteers are.
Excellent. Well, I have to say thank you so much, Shona. That was excellent and really, really fascinating. Thank you so much for joining us today. It's really been our pleasure to have you at the Orkney International Science Festival. Thank you.
Thank you so much. I really enjoyed the opportunity, and thanks to all the organisers for all the hard work you've put in.
Thank you. Thank you very much. Well, I'll have to do a little bit of housekeeping now, and I have to say, unfortunately, our next talk at 5 o'clock has unfortunately been cancelled. That's the Science Under Sail, and it was an event that was going to be broadcast from a live tall ship, the Pelican, but unfortunately they're in quite rough weather at the moment, and they're unable to make an internet connection. So we're sorry that has to be cancelled, but we'll see what we can do if, once the storm passes, if there's anything we can do then. So our next event will actually be at 6:30, and that'll be Song of the Whales with the St Andrews Sea Mammal Research Unit. Excellent. And of course, there are still available places at the PD Kirk lunch event tomorrow, and also at our Festival Club table sessions. So the links can be found in the chat, the live chat now, and also the descriptions on the website below each PD Kirk lunch event. And please come along. Please come along. If you're enjoying the festival, please like us on our Facebook page and please follow our YouTube channel. And thank you to everybody asking these great questions, and thank you so much for tuning in to us this afternoon. And a very, good afternoon and goodbye from all of us here. Excellent. Cheerio.